Hello and welcome to this video on section 22.5 on the formation and selection of T lymphocytes in primary lymphoid structures. So we know that our T cells are going to originate in the red bone marrow, and like we said in the last section, they then travel over to the thymus as our pre-T lymphocytes so that they can complete their maturation process. And initially, they're going to have both of the CD4 and CD8 proteins, and through the process that we are about to discuss, we will select one or the other. They'll also have their T cell receptors that are going to be produced randomly with variant genetic makeups, and each cell will have its T cell receptor tested through the process of selection, which again we'll discuss on the next slide. We'll see if it would bind to antigens or if it is going to bind to its self-antigens. And here we are. We are going to go through thymic selection, and that will eliminate a whopping 98% of the T cells that are produced. We do this through two different selections. First, we have positive selection, which you could see pictured over here. Our thymus, specifically in the cortical region, is where we will have positive selection take place, and we will have these thymic epithelial cells there present their MHC molecule to a thymocyte. Our thymocyte being the pre-T cell over here. And if they are able to bind, then they survive. So you can see kind of our schematic here. Does it bind? Yes, survives. No, then it's going to be destroyed through apoptosis. We also have the process of negative selection, where we are going to have take place in the medulla of the thymus. And this survival is dependent upon not recognizing our self-antigens. So, for instance, we would have our dendritic cell present that self-antigen on a MHC class 2 molecule here, as well as a MHC class 1 molecule. And if the thymocyte is going to recognize the self-antigen, then we are going to destroy it through apoptosis because we don't want these immune cells to bind and then attack it. Whereas if they don't recognize it, then we will allow that thymocyte to survive. And so this process really helps us gain self-tolerance, meaning we are able to tolerate our self-antigens. And once we go through all of this entire process, we will gain central tolerance, meaning that we went through the selection process within our primary lymphatic structures. Our next step is to allow those T lymphocytes to differentiate. So our helper T cells will lose their CD8 protein and keep the CD4, and our cytotoxic T cells lose the CD4 protein but keep their CD8 protein. And here is a selective loss of either the CD4 or CD8 protein. Like we said, we are only going to keep 2% of our T cells through the selection process. And when they're left, our helper T cells are going to have their CD4 proteins, and our cytotoxic T cells will keep their CD8 proteins. So our T cells then migrate from the thymus to secondary lymphoid structures. Remember, that will include things like the spleen, our lymph nodes, our tonsils, and they are immunocompetent, meaning they're able to bind to an antigen and be able to respond to it, to have an immune response, that is. So if they have not come into contact with an antigen yet, and they're just kind of hanging out in the secondary lymphoid structures, we call them naive T cells. And it isn't until we come into contact with an antigen that that cell becomes active. Let's talk about our regulatory T lymphocytes, also known as Treg. They also are going to have the CD4 protein on them and will be able to bind to self-antigens. Now, I know you might be thinking, wouldn't that trigger an autoimmune response, but we do need some of these regulatory T cells so that we can stop an immune response from taking place beyond the point of fighting a pathogen. So they're also functioning in tolerance outside of the primary lymphoid structures though. So this is why we call this type of tolerance peripheral tolerance. So this really comes into play when you are combating an infection. Let's say you have just a cold virus and you're really kind of suffering 2-3 days, and then you start to feel better. That's because the immune system has fought off that virus, kind of limited its spreading, and we are able to get rid of the cells that have been infected with viruses, and we want to start minimizing that immune response since we have combated the pathogen. So it is really the regulatory T cells that stops that immune response. Now, sometimes when we have tumors in our body, it can foster Treg proliferation, and this works to the tumor's advantage because it is going to inhibit our immune response, which stops our NK cells and our cytotoxic T cells from finding these cancer cells and killing them. And then we also have potential cancer treatments that are in development that involve diminishing the inhibitory influence of tumor Tregs so that we can allow our immune system to respond properly to these cancer cells.